My presentation will be on the use of open field tests as an assay of anxiety behavior in mice. My name is Andrea N. Rivera Velez, and I'm a subgraduate student at Dr. Velazquez's lab. Our goal is to see if bisphenol A, otherwise known as BPA, has a profound effect on mice's anxiety levels in an open field test. Bisphenol A is a chemical present in polycarbonate plastics and epoxy resins, which are used in food and drink packaging. Since plastic pollution has become an overwhelming concern, it is common to find BPA is in the air, dust, food, and water. However, we are primarily exposed to it through oral routes of administration when we expose this compound to high temperatures or pH changes, whether that be heating our food in plastics that contain BPA or being served hot food in these plastics. The temperatures cause BPA to break down and be released from the plastic. Thus, we ingest it along with our food. In 2004, the CDC found that 93% out of 2,517 urine samples had detectable BPA levels. Also, the Environmental Protection Agency places BPA at its third highest priority environmental hazard. Some studies have shown that the lowest acceptable BPA amounts have caused effects in the development of secondary sex characteristics and behavior. It can also cause cognitive impairment and increases in anxiety. Moreover, it has been seen to cause neurotoxic effects in the brain. Other than adding data which shows the effects of BPA in mice, we are doing this study because late last year and earlier this year, Chiara Cardona, a graduate student from our lab, found that when mice are injected with a BPA solution, they show deficits in learning through fear conditioning experiments. In this type of paradigm, a mouse is exposed to a shock that serves as a stimulus in a defined context. The next day, the mouse is inserted into the same context without the shock to see how much time the mouse spends freezing due to danger anticipation. As time goes by, the freezing should diminish. The day after, the mouse should show minimal signs of freezing when inserted into the context if they consolidated the extinction memory the previous day correctly because they recognize that the context no longer shows any danger. Our data showed that freezing decreased considerably in the experimental group when compared to the control group in both the conditioning session and the extinction session, meaning that the expression of fear has decreased. We will explore BPA's effects on anxiety through open field tests. The amygdala is an important area in the brain for detecting potential threats or dangers and further mediating anxiety. It has been reported that a group of neurons in the basolateral amygdala fire under anxiety conditions in both open field and elevated plusmates tests. The open field test can measure anxiety by quantifying the amount of time a mouse spends around the edges of the field or in the center of the field. Since mice show anxiety-mediated fear responses to open, brightly lit environments, it is expected that they will spend more time around the edges of the field at the beginning of the test, and then as time passes, more time around the center of the field. But this will only occur if their anxiety is reduced. If it isn't, we will see high levels of time spent around the edges of the field. We chose open field tests because a number of important conventional and etiological parameters can be collected and analyzed during the performance, such as behaviors ranging from overall locomotor activity to anxiety-related emotional behaviors. Context fear conditioning and extinction tests mainly affect hippocampus-based learning and involved basolateral amygdala-associated circuitry. The open field tests mainly addresses effects to the basolateral amygdala circuitry in response to BPA exposure. By comparing the results from these paradigms, we will be able to narrow down where BPA mainly affects anxiety-related behaviors. Previous studies have shown that at low BPA concentrations, there are some neurotoxic effects in the brain and behavior changes. Animal studies have linked this chemical to anxiety, aggression, poor learning, and social behavior. Early developmental exposure to BPA produces increasing sensitivity to stress, the risk of developing anxiety behaviors, and increases the FKBP5 gene expression in the brain, which modulates stress response. Our hypothesis is that if relevant doses of BPA through oral exposure for 2 weeks in young adult mice have profound effects, then these mice will show high levels of anxiety. We have seen deficits in extinction learning through fear conditioning and extinction paradigms, and now hypothesize that open field tests will show us deficits in mice anxiety levels. Our methodology is quite simple. We plan to set up an open field testing area, which will be comprised of boxes measuring 50 centimeters by 50 centimeters and at least 38 centimeters tall. Then we will administer saline intraperitoneally to our control group and BPA injections intraperitoneally to our experimental group. We will place these mice in the open field and measure their response. Afterwards, we will analyze the data that we've collected. The following video is a generalized concept of what an open field test is.
Welcome to Maze Engineers, Maze Basics, Open Field. The open field test utilizes a box-shaped apparatus. Though the apparatus looks simple enough, the test is pretty clever. Rodents naturally have an aversion to open areas because open areas leave them exposed to predators, but they also like to explore new areas. This conflict allows us to measure their anxiety levels as well as locomotor and exploration activity. There are a variety of things you can measure to get a sense of an animal's anxiety levels. A common measure is the amount of time the animal spends walking along the outer edge versus the center. The more anxious the animal is, the more time it will spend on the outer edge. This test is also useful for looking at the effects of drugs such as anxiogenics and anxiolytics, which cause and reduce anxiety respectively. The anxiogenics will cause the mice to spend a larger amount of time on the outer edge of the apparatus. The anxiolytics, on the other hand, will reduce the anxiety-like behaviors and lead to a fairly even distribution in the amount of time spent in each section. The open field strength lies in the fact that it can be paired with other anxiety measures, such as the elevated plus maze, Barnes maze, and zero maze. It can also be paired with the novel object recognition test and serve as both a habituation time to the apparatus as well as a second measure of anxiety.
As we saw in the previous video, we can say that if a mouse spends more time in the inner zone than in the outer zone, and it was in the BPA experimental group, then the BPA had anxiolytic-like effects on these mouse. But if we see, when we compare the results of the experimental group to the control group, that the mice in the experimental group spent more time in the outer zone than in the inner zone, that we could say that BPA had anxiogenic-like effects. As we have mentioned before, we hope or we hypothesize that this is what will happen. And when we obtain these results, we will compare them to our preliminary data, which was the fear conditioning response experiments, and we hope to be able to pinpoint exactly what areas in the brain BPA affects when it comes to anxiety. That has been all for today. Thank you for your attention. I hope you enjoy the other presentations and have a nice day.